Hi friends, in this video we are going to be having a discussion about a very important and an upcoming topic in the area of oncology, which is given the name of liquid biopsy. So herein we start our discussion of the concepts of liquid biopsy. Now whenever we talk about liquid biopsy, I just want you to understand that this is an example of a non-invasive method which is useful for making a molecular diagnosis of cancer. Whenever we are talking about this kind of a technique, it is based upon the detection of certain types of biomarkers which are present in the different types of body fluids. I want you to understand that tumor cells are responsible for release of certain biomarkers, and if we are able to detect these biomarkers in the body fluids, that is helpful in picking up a cancer easily. Because you are using over here any kind of a body fluid as a sample, which is an example of a liquid, so that is why the term used over here is liquid biopsy. The first potential thing which any examiner may ask you subsequently is, what are the kind of body fluids which can be used as a sample over here? So what we can be using in these patients can be a sample like blood. We can be using in the sample something like plasma. We might as well be using a sample like urine. We can be using a sample like cerebrospinal fluid. You can be using bronchiolo-alveolar lavage fluid. Might as well use ascitic fluid, or at times uncommonly you might as well use even something like the breast milk. What exactly are the types of the biomarkers which can be released by the tumor cells? In order to understand this, I would just like your attention to be focused on the screen, because I want you to understand as a concept that, let me say, this is an example of a collection of tumor cells. It can be a primary tumor, it can be a secondary or metastasis. Whenever you're going to be having a tumor, some cells in the tumor are going to be more aggressive in nature. So these cells are going to penetrate the vascular wall, and these cells are going to appear in the systemic circulation. Such example of cells are being given a name CTCs, or circulating tumor cells. Fact number 2, we can be having a situation wherein the tumor cells are responsible for active secretion of nucleic acids, or they undergo the processes of necrosis or apoptosis. Now, whether by secretion, whether by necrosis or by apoptosis, if there is going to be a release of nucleic acids from the tumor cells, the nucleic acids can enter into the systemic circulation. Majority of the times it is the DNA which is being released from the tumor cells. So this kind of a DNA is being given the name of cfDNA, or cell-free DNA. Fact number 3, sometimes the tumor cells are responsible for causing secretion of membrane-bound vesicles, which are being given the name of exosomes. These exosomes are actually going to be containing the presence of DNA. They may be having RNA, they can be having proteins. If you get either of these in the blood sample of an individual or the body fluid sample of an individual, you can extract them. You can do a subsequent genetic analysis on them, and you can find out what exactly is the kind of the translocation or the gene mutation which is present in the tumor over here. And that is how you are going to be making a diagnosis of a particular tumor. So since you are taking a body fluid over here and detecting the presence of a biomarker, so this technique is being given the name of liquid biopsy. Now, how is a liquid biopsy going to be different from the standard tissue biopsy that we talk about or that we perform routinely? I just want you to understand as a concept that whenever you're talking about a standard tissue biopsy, it's an example of a more time-consuming procedure. You can be having a situation where you are inserting the needle only at one particular site, and therefore you're going to be having a localized sampling of the tissues. Sometimes the tumors are not very much approachable anatomically, so it is not easy to obtain a standard tissue biopsy. You might as well require the help of the radiological guidance like CT scan or ultrasound guidance. We need to have a knowledge about the fact that this procedure is going to be invasive, and therefore procedure related complications as simple as something like pain is going to be present in the patients. We need to understand that sometimes the surgeon has performed a surgery and therefore the main tumor has been removed, or the tumor is very small in size after the administration of chemotherapy or radiotherapy, and therefore it is not possible for you to detect the presence of a primary tumor for the standard sampling. Another important problem that we find is that sometimes there is presence of the concept of tumor heterogeneity. For example, suppose you have a patient who is having a lung tumor, and this lung tumor now metastasizes, let us say, to the adrenal gland or to the bone or to the brain. At the site of metastasis, the tumor cells acquire more mutations. So there was an initial or a primary tumor which was already having a mutation. At the site of secondaries or metastasis, tumors acquire more mutation. If you are going to be performing a standard tissue biopsy, you are taking the tumor cells from only one site. So you will not be able to detect the genetic mutations present at the other site. Therefore, I have written over here that tumor heterogeneity cannot be detected by standard tissue biopsy. Compare and contrast this with what we are going to be finding in case of liquid biopsy. This is an example of a procedure in which you are taking a blood sample of an individual. Therefore, it is very easy to perform. Therefore, this is going to be an example of a procedure by which genetic analysis is done in a short span of time. So results are quick. You understand the fact that you are going to be having a comprehensive tissue profile. It's easier for you to obtain a sample. We need to understand the fact it is a minimally invasive procedure. So procedure related complications are going to be very less. We can have the presence of a situation wherein we have a minimal tumor load after maybe a treatment or after a surgery. So even in the situation where you are going to be having a minimal tumor load, even then you can be detecting the presence of the mutated cells over here or the mutated nucleic acids. Tumor heterogeneity can be detected easily because from the primary site there will be some tumor cells which are released. From the secondary site there will be some tumor cells which are released. You can filter out both of them, do separate genetic analysis for them, and you can get to know what exactly are the genetic mutations present at either of these sites. So this tumor heterogeneity can be detected. Now, because standard tissue biopsy is an example of an invasive procedure, so if the patient has to undergo the testing 3 times or 4 times so as to monitor the treatment, then it becomes cumbersome for the patient. Whereas liquid biopsy is going to be something in which repeated testing is easier because it is as simple as taking the blood sample of the patient. The only disadvantage that we have in context of liquid biopsy is a fact that in these patients you are not going to be having the presence of a large number of tumor cells. So you kind of need to amplify whatever little material that you are getting from the blood or the body fluid. Fact number 2, we are in the process of actually developing standardization techniques for making a detection of different types of tumors. So this is yet which is going to be in a nascent stage, but gradually over a period of time we anticipate that in the coming years we are going to be having the liquid biopsy as one of the major methods for making a diagnosis as well as deciding the disease progression in the patients. We are now going to be having a discussion about the different important types of the biomarkers that we just made a mention of. The very first example of a biomarker that we need to have an idea, friends, happens to be what is given the name of circulating tumor cells or CTCs. I just want you to understand the fact that whenever you're talking about tumor cells, they are going to be very less in number in comparison to the normal cells in the body. So let me just give you an idea with respect to the fact that if you're talking about the normal WBCs, the normal WBCs are going to be in the range of, as most of you are familiar, maybe in the range of somewhere close to 10,000 cells per microliter. Whereas whenever you're talking about the circulating tumor cells, they are going to be present in far less number. The important point that we need to understand is that the tumor cells which are going to be appearing in the circulation are the ones which are going to be more aggressive. So we need to look at the genetic material that is present in these circulating tumor cells. Since they are less in number, so we have to use special techniques by which we separate the circulating tumor cells from the normal cells in the body fluids. And for that very purpose, we are using the techniques which are given the name of enrichment of CTCs. This is dependent upon 2 important properties. One can be the biological properties of the CTCs, or it can be dependent upon the physical properties of the circulating tumor cells. Let me just help you with that simple diagrammatic representation. Let me say this is an example of a circulating tumor cell, and this tumor cell is going to be having the presence of an antigen. On the other hand, let me say this is an example of a normal cell of the body, normal WBC. This is also having the presence of an antigen. What you can do over here is you can have the presence of an antibody which will attach itself with the antigen present on the surface of tumor cell. If I am using a special tagged antibody which attaches itself with the antigen on the surface of a tumor cell, I will be able to separate out only the tumor cells. So out of a cluster or group of cells, if I am able to separate the tumor cells only, this is a process which is given the name of positive selection. So out of so many cells, I take out only the tumor cells with the help of tagged antibody. The only disadvantage that we talk about with respect to positive selection is that you must have an idea about the kind of antigens expressed on the surface of tumor cells. Now that is not something which is very easy. So what we follow is we are going to be looking or doing the process of what is given the name of negative selection. In negative selection, what you understand is that the normal WBCs are going to be having the presence of a molecule given the name of CD45. we actually ensure that there is an antibody against CD45. If we have a tagged antibody against CD45 and this is mixed with the sample, well, what will it do? It is going to attach itself with the normal WBC. So all the non-attached cells are going to be the tumor cells. So the tagged antibody is going to be associated with normal cells and the unattached cells are the tumor cells. So that is a process that is given the name of negative selection. It is always better that we go in for something like negative selection because that eliminates the selection bias and that is going to be more simpler for you, as it is not possible for us to know about each and every antigen present on the surface of a tumor cell. The same thing is what I am going to be depicting over here with the help of this kind of an image. So I am just trying to show you over here that these are example of tumor cells which are having the presence of an antigen. One of the classical antigens that we are using currently happens to be what is given the name of EpCAM, which is epithelial cell adhesion molecule. So if you have an antibody against EpCAM, it is going to be able to pick up the tumor cells. The other thing that you can do is negative selection, in which you take an antibody directed against CD45 antigen. It is able to pick up the non-cancerous cells and therefore it is given the name of negative enrichment. Once you are going to be having the knowledge of the biological property, the second property by which you can distinguish or enrich the circulating tumor cells is physical property. The circulating tumor cells are going to be bigger in size and therefore you can have different types of techniques based upon size. So you can be using something like the different kinds of filter. You can be using a technique in which you are going to be having the presence of different kind of density gradient media, or you can be having a process that is given the name of dielectrophoresis. So any of the physical properties which distinguishes a normal cell from a circulating tumor cell is being used, and now you are going to be having the availability of the circulating tumor cells. Once you have the tumor cells available with you, now that they are going to be more in number, so what you can be doing is you can be extracting their nucleic acids, and these nucleic acids are going to be subsequently going to be processed by different techniques. Say for example, you can be using something like immunostaining, or what you can be going in for would be maybe using the circulating tumor cells fluorescent in situ hybridization. You can be going in for something like the polymerase chain reaction. For quantification, you can be going for something like quantitative real-time PCR. You can be going in for something like sequencing. As a lot of you, I believe, have an idea about the basic fact with regards to sequencing, the gold standard happens to be what is given the name of Sanger sequencing. You have the other modified variants like pyro sequencing, and currently what is being done, because you want to process a greater number of the gene sequences, we are having what is being given the name of the next generation sequencing. Availability of circulating tumor cells also helps you perform what is going to be given the name of a cell culture. So once you are going to be having the presence of the circulating tumor cells, all these molecular techniques can be performed for the genetic analysis, and thereby you can find out what exactly was the problem in the tumor cells, what kind of mutation was present, what kind of chromosomal defect was present, what kind of copying number variants was present. So all these things are going to be telling you about the problem that was seen in the cells as such, and the same thing is what I am trying to show you with the help of this kind of a simplified flowchart. So you have taken the blood sample of an individual, you have enriched it with the technologies based upon the biological or the physical property, and subsequent analysis done with the help of immunostaining, FISH, sequencing, the quantitative real-time PCR, or something like cell culture. That is what we need to have an idea so far as the circulating tumor cell as a biomarker is concerned. The second important example of a biomarker that we are using in context of liquid biopsy is what is being given the name of the cell-free DNA or the cfDNA. Now, as we talked about the fact that whenever you're going to be having the tumor cells can be directly responsible for the secretion of nucleic acid material, or there is presence of necrosis or apoptosis in them, because of which a cell-free DNA is going to be released by the tumor cells. Now this kind of nucleic acid material that is going to be released over here is what is going to be given the name of the circulating DNA or the circulating nucleic acid material. Now whenever we are talking about the circulating DNA, this can be of two types. The first situation can be that you are going to be having the presence of a cell-free DNA. So this is something that is going to be seen in case of a normal situation. That means normal cells can be having a secretion of the cell-free DNA, and there can be situations wherein in pathological processes also you can be having the presence of cell-free DNA in the systemic circulation. One of the special subtypes of cell-free DNA that we need to have an idea is given the name of ctDNA. Now, whenever we are talking about ctDNA, CT stands for circulating tumor DNA, and I want you to understand certain basic concepts over here. ctDNA is a special subtype of cfDNA or cell-free DNA. Why do I say so is because this is an example of a circulating or cell-free DNA which is going to be secreted by the tumor cells themselves. It's important for us to understand the simple concept that whenever you are going to be having the circulating tumor DNA, the amount of circulating tumor DNA is going to be directly proportional to the number of tumor cells that are present in case of a patient. So whenever there is going to be higher tumor load in an individual, there is going to be more secretion of the circulating tumor DNA. And when we get the circulating tumor DNA, this is something which can be diagnosed or this can be analyzed to see for the presence of the specific mutations which are present. Now the process by which you are going to be processing the circulating DNA or the circulating tumor DNA is what I wanted to understand. So you take the blood sample of an individual, and please remember this is where the areas of the MCQs are going to be loaded with. So the blood sample of this individual is going to be taken in a tube which is going to be containing tripotassium EDTA. A lot of you may be having an idea. I just want you to not miss out on that question. The EDTA containing tube is going to be having a lavender top. So that can be an image based question, that can be a direct theory based question for the exam. Remember the fact that whenever you are talking about the ctDNA or the circulating tumor DNA, this DNA is relatively unstable. It's having a short half-life. So the half-life of this kind of a DNA is going to be varying anywhere between roughly 15 minutes to 2.5 hours. Therefore, once you have taken the blood sample in the EDTA tube, this blood sample has to be sent to the lab and the plasma has to be separated at the earliest. This plasma separation has to be done, and that is another potential question. Please remember, this has to be done within 1 hour of receiving the sample. Now, since the nucleic acid material is unstable, so we need to keep this plasma safe, and this plasma has to be stored therefore at a temperature of minus 80 degrees Celsius. So I want you to remember and understand the fact that you need to be familiar with the short half-life of the circulating tumor DNA. You need to be able to pick up the tube in which the sample is going to be taken. You should be having an idea what is the time within which the sample has to be processed, plasma separation has to be done, and the temperature at which plasma has to be stored, minus 80 degrees Celsius. Once you are going to be having the extracted sample, then the subsequent analysis of the sample is what is to be done. Now this analysis of the sample is being done in two approaches, one which is given the name of a targeted approach and the other one which is being given the name of a non-targeted approach. The simple concept is, if you already know what kind of genetic mutations are you expecting, then you go in for something like a targeted approach for the genetic analysis, something that a lot of you may be having an idea. We go in for something that is given the name of digital polymerase chain reaction, or you are going to be going in for something like the real-time PCR, or you are going to be going in for something like the targeted next generation sequencing. If on the other hand, I have no idea about what kind of mutations or abnormalities am I expecting, I will have to search for the complete genome, and therefore this is what is going to be given the name of the whole genome sequencing. Whole genome sequencing. So depending upon what exactly is going to be the expected trouble or the unexpected mutation that you are having, you are going to be choosing a particular technique. Just to give you a couple of examples in the clinical side, suppose you have a patient in whom you are looking something at a non-small cell lung cancer, adenocarcinoma of the lung. So you would go and check out for the presence of something like the EGFR mutation. So that can be an example of a targeted approach. Similarly, in a patient who is having malignant melanoma, you can check out for the presence of something like BRAF mutation. The same thing is what I've tried to depict over here with the help of this kind of a workable flowchart. So I don't want you to miss out on the important points over here. So blood sample is supposed to be taken, and the tube which is being taken over here is lavender color tube which contains tripotassium EDTA. The sample has to be spun within 1 hour of receiving it. The plasma has to be stored at 80 degrees Celsius. Sample being extracted because of the unstable nature of the circulating tumor DNA, and then subsequent analysis which is being done by either a non-targeted approach, the whole genome sequencing, or by the targeted approach, digital PCR, or the targeted next generation sequencing. Be familiar with the fact that currently we need to have an idea that sometimes it is not possible for any individual to collect the sample and send it to the lab very early. So what we are having over here is, nowadays we are having the availability of what are being given the name of preservative containing tubes. The preservative containing tubes are going to ensure that the circulating tumor DNA in them remains stable for a longer duration of time. And if you are using a preservative containing tube, this is going to ensure that the cell-free DNA or the circulating tumor DNA remains stable for 96 hours at room temperature. But it is ideal that once the sample has been collected, send it to the lab as early as possible for the subsequent analysis. Be familiar with the fact that the classical nucleic acid sample that we are using over here is a circulating tumor DNA. But with our advancement, we understand the fact that nowadays we are also having tools by which we detect or analyze the presence of microRNAs. I do expect a lot of you who have already seen the videos in neoplasia to have a basic idea about microRNA. MicroRNAs are non-coding RNAs which are responsible for RNA silencing by the use of the dicer enzyme and the RNA-induced silencing complex. These microRNAs are responsible for causing a change in the functioning of the different RNAs, and therefore currently they have been implicated in the development of many pathological conditions including cancers. So, if you are going to be having a detection of a particular microRNA, this can be useful for making a diagnosis of the specific mutations in certain cancers. Recall the fact or understand the fact that the sample which is preferred for microRNA analysis is usually serum. Because as a concept, it is said that if you are taking plasma, you can have some contamination of the RNA coming from the platelets at the time of coagulation. So serum is a better sample. That can be an advanced question. Fact number 2, whenever we are talking about microRNA, we are going to be using the specific microRNA quantification, which can be done with the help of a technique like quantitative real-time PCR. So just to help you recall what we discussed in the videos in neoplasia, I would help you remember the fact that we talked about something like MIR 15 and 16, and those were implicated in the development of conditions like B-cell lymphoma. So that is what we need to have an idea so far as the second biomarker is concerned. The third example of a biomarker that we need to have an idea is a component that is given the name of exosome. Now I do believe the fact that you may have heard about exosome in your class 11-12 biology classes as well. Exosomes are actually example of membrane bound vesicles. Now these membrane bound vesicles are actually going to be having the presence of nucleic acid material like DNA, RNA, and there is presence of the proteins as well in them. Just recall the fact that when you are talking about the formation of an exosome, this is actually an example of a structure derived from an endosome. So if you are following over here, the first thing that you have is process of endocytosis. This is going to result in the formation of an early endosome. When this early endosome is being produced, there is going to be an entry of different kind of nucleic acids as well as proteins from the cytoplasm of a cell, and now it is going to be resulting in the formation of a late endosome. First possibility is that a late endosome fuses with lysosome and is going to get degraded. The second possibility, however, is that this late endosome resists the degradation and now it is being thrown out of the cell. So what will that result in? You will be having a membrane bound vesicle which contains the DNA, RNA, lipids and proteins, and because it is exiting from the cell, so exosome. Now this exosome is going to be released from the tumor cell and it is going to be having its interaction with the recipient cell as well as the immune cell. So this can function as an intercellular messenger. What is the interaction and what is the importance of knowing about this interaction? When this exosome is going to interact with the immune cell, it reduces the activity of the immune cell against the tumor cell. So that helps in more aggressiveness or more easy spread of the tumor cells without being killed. Fact number 2, exosomes are going to result with an interaction with the recipient cell. So what has been seen is that they are responsible for causing a transformation of a normal cell of the body into a malignant phenotype. So that is why I said that this is important for intercellular messenger. Now whenever we are talking about an exosome, this diagrammatic representation will help you, and that is exactly what I have talked about over here with you. So potential questions for the exam that you need to have an idea: endosomal origin, the size is what they may ask you maybe in an advanced question, 40 to 100 nanometers. This is going to be very important as an intercellular messenger. I want you to remember the fact that in a lot of tumors, the formation of exosomes actually is going to be upregulated, and these exosomes are going to be now available or present in the body fluids. So classically they can be present in maybe something like blood, they can be present in plasma, they can be present in cerebrospinal fluid, or that can be present in the bronchiolo-alveolar lavage fluid. We need to understand the concept, which is that the RNA that is present in an exosome is extremely resistant to the standard RNases that are present in the body. Why that becomes significant? If this RNA is going to be resistant, that means it is going to be more stable. If it is more stable, that means it is going to be useful for better detection, as it will be having higher sensitivity as well as specificity. So this RNA that is available in case of an exosome is going to be useful, and this is going to be analyzed further to detect the presence of the specific defect or the mutation which may be present in a particular tumor. So those are the important words that we need to remember so far as an exosome is concerned. The fourth and the most novel marker that we are having currently in context of liquid biopsy is what I have written over here for you friends. This is given the name of tumor-educated platelets. Now, a lot of you may have watched the videos on the genetic basis of carcinogenesis, and I just want you to recall that simple concept. It is said that now for the progression of a tumor, even certain platelets are going to be having a very important role. Why do we say so is because of a simple fact. Platelets contain lot of growth factors, including the important one, something like the platelet derived growth factor. Because of these growth factors, the platelets are responsible for the processes like invasion of a tumor as well as for more blood vessel formation or angiogenesis. The platelets are responsible for secretion of these factors, and these factors are responsible for a very important transformation process given the name of epithelial mesenchymal transition. So because of all these factors, what we understand is that there are certain platelets which have been educated by the tumor cells to secrete factors, and they are going to be responsible for causing more aggressiveness or more invasion of the cancer. So we can have platelets as a sample, and the RNA or the nucleic acid material present in the platelets can be sequenced. This is useful for differentiating between the normal platelets and the tumor educated or the abnormal platelets. And that genetic material sequencing or analysis can help us know what exactly is going to be the problem that is going to be seen with a particular malignancy. So that is what is going to be the most novel biomarker apart from the three that we talked about earlier, apart from circulating tumor cells, the circulating tumor DNA and exosomes. We therefore understand the fact that if I have to talk about as a summary the uses of liquid biopsy, these are the summaries of liquid biopsy that I wanted to understand as a concept. Screening of a cancer, which means this is going to be useful for making an early detection of a cancer. This is going to be useful because the liquid biopsy can help you pick up the recurrence of a cancer. And this is going to be also important because certain mutations might as well tell you about the prognosis of a patient. The easiest example I can share with you over here with respect to the diagnosis of a cancer is, say for example, if you have a patient who is suffering from something like non-small cell lung cancer or adenocarcinoma of the lung. These patients are going to be having the presence of epidermal growth factor receptor mutation. So that can be useful for picking up a cancer like this. Similarly, we can be having something like the detection of various problems in patients having malignant melanoma, very useful for patients having breast cancer, patients having colon cancer and pancreatic cancer as well. What we understand is that this can be useful for the purpose of disease monitoring. And when I talk about disease monitoring, I want you to understand, suppose there is a patient having a cancer in whom we start the treatment. Initial phase, the patient responds very good. But after some time, the response of the therapy is not going to be good. This is because of development of a phenomena of drug resistance. And why will drug resistance take place? That is because of the fact that tumor cells acquire secondary mutations. So what are these secondary mutations? That is again something which can be picked up with the help of liquid biopsy. Third fact, we can be using liquid biopsy for enabling us the targeted treatment of a cancer. Just to cite you the same example, suppose you had a patient having adenocarcinoma of the lung and the patient was having epidermal growth factor receptor mutation. So I can be giving specific tyrosine kinase inhibitors, if you can just relate it with your horizontal knowledge of pharmacology, a drug like erlotinib, which can be useful for treatment of this patient. More importantly, as a research and development area, suppose the patients are going to be having the presence of a mutation. So we can know a new target so as to develop a new drug which can be useful for this special category of the patients. All in all, what I can tell you over here is that liquid biopsy is currently a very important revolution in oncology, and that's a summary that I've taken from the journal Nature to help you just recall the fact that whenever we are talking about patients who are going to be having a cancer, you are going to be taking their blood sample or body fluid sample. You are going to be extracting circulating tumor cells or the circulating tumor DNA or exosomes, the circulating tumor DNA is going to be analyzed for presence of translocations, for presence of chromosomal defects, for deletions or insertions, for epigenetic markers, point mutations, or certain amplifications. If I'm talking about CTCs, since they are less in number, so they have to be enhanced, and the enhancement is dependent upon either the biological property, positive and negative selection, or the physical property dependent upon the size, the filtration, the immunodiffusion, etc. We are then going to be having the CTC detection using the immunological and the molecular biological techniques. And finally, once you have got the CTC, you analyze it with respect to the genome and with respect to the proteome. That means you are going to be looking at the genetic markers or the molecular analysis, and you're going to be looking at the protein molecules which may be present over here, and that is what is going to tell you about the nature of the specific mutations which are going to be seen with a particular cancer. So all in all, a very, very high topic for the exam, and definitely a very exciting area with respect to oncology. So this was our discussion of the concept in the video that I had talked about over here, friends. Please do press the like button, do subscribe the channel, give your valuable comments to let me know what are the other topics on which you want me to make those videos for your better and conceptual understanding of the subject, so that you love the subject all the more. Best wishes.